This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 66, recorded on January 8th, 2014. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Good afternoon, Vincent.
Happy New Year.
And to you.
It's our first Your and Mine, first podcast of 2014.
So it is.
So what are your feelings about 2014?
Well, I just hope it's better than 2013. Yeah, no, I always look forward to a better year, even though sometimes you don't get it. You have to anticipate the best.
Did you have a bad year in 2013?
No, I thought I had a very good year.
As far as I can tell, you were very active.
I had a good time. I went to some interesting places. In fact, I just got back from an interesting place. So I spent not half, but I spent part of last year in a place and part of this year in the same place.
Oh, by the way, how was New Year's in India?
New Year's in India was interesting.
What city were you in?
We were in Madras.
No, they don't call it Madras anymore.
It's not the city we were in anyway. We were in Madurai.
What's the current day name of Madras?
The current day name of Madras is Chennai.
Chennai. You were in where?
We were in a city south of there called Madurai.
And did you celebrate the New Year?
We tried.
What do you mean we tried? You fall asleep?
Yeah, we did, actually. We had a nice meal. We had a few beers, in this case, Kingfisher beer, which is their very good beer, right? And there's an airline.
I like Kingfisher.
There's an airline named after that, too, by the way.
This must be a regional airline, right?
It's a Kingfisher airline, Kingfisher beer. It's owned by the same people.
It's an Indian company?
It is an Indian company. And
it's not British, right?
No, it's not British. So we sat on the roof of the hotel that we were staying at, and we watched some fireworks for a couple of hours, and then we just got too tired and had to go to bed. So 2014 appeared
without you knowing
in our
it.
Dreams.
You woke up and it was 2014.
That's right.
I like 2014. It's an even number.
Agreed.
And
do we know what year it is in terms of Chinese things?
I don't know.
Like last year was, I think, the snake.
6,492,000,000. I just don't know.
No, we could look it up, of course.
Oh, you want to know what year it is?
Yeah, I would like to know what animal we're looking forward to being. I think someone said the dragon, but I think we've had that recently. So we've had rabbits and we've had snakes. So I don't know what else the Chinese New Year's will bring to us, but in New York City, they celebrated a very nice New Year's celebrations down in Chinatown, and I've actually been in Chinatown when they were setting off their fireworks, which is it's legal for them, but not so legal for us.
I searched this year in China, and it
no, not China. The Chinese calendar, just a Chinese calendar.
On the same page is one of my blog posts. How about that?
Yeah,
I'm popping up everywhere.
How did that work?
It's about a polio in India, but
I sent you a picture.
What did you want me to say? Chinese
New Year.
New Year. Yeah, I tweeted that picture. It's very nice.
Good.
And I put your name on it.
That was in Uti.
All right. Chinese New Year, is that what? So it's actually, it's not till January 31st.
That's correct. And so what animal are we looking forward to being this year?
Well, let's see. Kung Hei Fat Choi. No, that's a film.
Uh-huh.
I think Kung Hei Fat Choi means Chinese New Year.
Could. I think we're losing the drift of the argument here.
Well, first of all, this is This Week in Parasitism.
That's right.
Now, here we go.
Aha.
2014.
Yes.
Horse.
Year of the horse.
You know, that repeats. The last time it was horse was in 2002.
Got it.
Last year was the snake.
Right. That I knew.
Next year is the goat.
Right.
How do they pick these animals?
I have no idea.
Okay. Do you have any listeners who know?
I'm sure there are some out there that would have some information on this, any more than they would probably have information on the calendar with regards to the zodiac.
Right. All right, we have a paper today that you selected.
I did.
It's not even out yet. It's a preprint.
That's correct. This is pretty cutting-edge stuff.
And it is in a journal that is called Antimicrobial Agents and Chemotherapy.
Right.
And the title is Reversible Cysteine Protease Inhibitors Show Promise for a Chagas Cure.
Correct.
And it is from McGill and Merck Frost.
Right.
Merck Frost is I guess it doesn't exist anymore.
They're dedicating the paper to the demise of that particular institution.
I find that weird. they have on the front page, this paper is dedicated to the memory of the Merck Frost Center for Therapeutic Research, which was closed in October 2010.
Right.
To the memory of? There's no memory of a Merck Frost. The people within it, maybe.
Yeah, well, they
sorry.
feel sad. I'm sure these people feel sad that doesn't exist anymore.
Well, Merck shut it down. Why are they dedicating it to something that the company shut down? It's just weird.
I don't know.
I tell you, all the Merck people, including my wife, who were laid off, 8,500 of them in the last weeks of 2013, they're not dedicating anything.
No, they're angry. I would be if I got laid off, too.
So can we, before we talk about this,
let's
Chagas disease,
let's
if I remember,
some of the biology here.
Trypanosoma cruzi?
Correct.
And Trypanosoma, no,
that's...
There's not another genus species?
Well, there are trypanosomes in Africa, and there are trypanosomes in America. So the
cruzi is American.
It is.
What is the African one? T.
Brucei.
Brucei, right, of course.
But it's not T. Brucei only. It's T. Brucei gambiense and T. Brucei rhodesiense.
All right. And in Africa, it's spread by sandflies?
No.
Black flies?
No.
Reduvid bugs. That's South American. South American is the reduvid bugs, right? They bite your face, so that's why it's called kissing disease.
Well, kissing bugs. Yes, that's right. You're doing okay. I mean, you're
Reduvido.
Your knowledge of vectors is good, but you just have misplaced the correct vector for the African trypanosome, which is an easy one to remember because it's got a double name. It's spelled the same. The double
Loa.
No, but you're getting closer.
Well, I should just look it up on TWiP.
No, it's Tsetse.
Why didn't I remember?
TSE.
I'm not myself.
You've been a little dizzy lately, Vince. I'm worried about you.
Don't worry, I'll be fine. No one will care when I'm gone except my family.
Stop it. I will care. Trust me. Because I don't even have to operate all this electronic equipment I'm looking at right now. What will we do without you? We need you. You're an integral part.
No, I am not.
You ask all the good questions.
But it's fine. It was here and it went. That's the way things are. Nothing's eternal.
But you know what? That's not absolutely true, though, because if we broadcast over iTunes and people listen to it, the sound waves travel outside of the atmosphere of the Earth and they continue on forever.
Yeah, but eventually they get so weak that nobody can
doesn't matter.
do anything with them.
It doesn't matter. We're producing sound waves that will forever reverberate throughout the universe as we know it.
Anyway, I have been dizzy.
I know you have.
Yesterday and today. I don't know why.
I'm concerned.
I have no infection.
You don't know that.
I have no symptoms except
you do
dizzy.
have a symptom. Well, that's a symptom. I hate to say it, but that's a symptom.
I have no respiratory symptom.
Do you know what the difference between a sign and a symptom is?
A sign and a symptom.
Sure.
A symptom is indicative of something.
Well,
whereas a sign tells you where to go.
Oh, dear.
No good?
Dear. No, that's not even close, Vincent. Not even close. So a sign is something that the doctor can see. Okay? He looks at you.
Oh, you're talking about medically.
Yeah, but of course. So if you were dizzy and you came to a physician and you said, I don't feel so good, and you said, I feel dizzy, what would you think they'd say to you?
Have you been drinking?
No, they would know that right away.
Have you had any infections lately?
That's right. Has anything hurt? Does anything hurt?
Have you taken any medications?
Right. Keep going.
Would they ask you if you took any drugs that are not licensed?
You, how long have you felt this?
How long have you felt this?
And what is the answer?
All my life. 2 days.
No. But then they would ask you to get up.
Yeah, and I would
try to walk.
I can walk; it's no problem. But
if you turn your head and try to walk, you said you get dizzy.
Yeah, when I move my head, that's
so you'd
when
say
it
that
happens.
to the doctor. You'd say, "Look."
When I change my glasses, that makes me get dizzy too.
Really?
So it's related to eye focusing.
So the sign for the doctor that you get dizzy would be that he would make a straight, or she would make a straight line, and and then ask you to walk that straight line. And you could do it without moving your head. But if they say, okay, now try moving your head and walking into the straight line, and you're going to get dizzy and you stop. Okay? So that's a sign. The symptom is what you feel.
Wow. So this is valuable for TWiP, right?
It is because we're talking about Chagas disease.
What's the sign of Chagas disease?
Thank you, Vincent. It's called Romagna's sign.
Romagna?
Romagna. Go to the internet, type out Romagna's sign on Google Images, and you won't believe the number of pictures you're going to get for this one. R-O-M-A-N-O. Romagna. L-O-N-A-S.
I'm sorry, how did you spell it?
Roman. R-O-M-A-N-A-S. Romagna's sign. Romagna.
R-O-M...
A-N-A.
Romana. Are you sure it's Romana?
Romagna. It's got a tilde over the end.
Oh, it's swollen eyes.
Yeah, eyes or eye.
One eye.
Unilateral periorbital edema.
Did we... we didn't mention this on our TWiP.
We're going to talk about it now. That's what we're doing now.
This is so incomplete.
No, that's not true. I think if you go back to the Chagas episode, we did discuss this, but only briefly.
Yeah, so every one of these people has one eye swollen.
They do, and in fact, when I was at the...
Is that a sign of Chagas?
That's a sign of Chagas.
All right, so let's write this down. A sign, because I need to put it in the show notes.
A sign is something that the physician can see, or your friend can see, or your parents can see.
Say it again.
Romagna's. The tilde is over the last N.
Which is periorbital, unilateral.
Unilateral periorbital edema. Mm-hmm. That's right. So as you look at a patient, usually it's a young person. Those are mostly children that are shown here. Okay? Mm-hmm. So it's interesting that this is an ancient known sign. How would we know?
As opposed to ancient unknown signs.
This is correct, because I was at the Anthropological Museum in Mexico City, which is a fabulous place to go visit. And there are pre-Columbian and post-Columbian. And when they say Columbian, what do they mean by that?
Colombia, South America.
No.
Columbus, Christopher Columbus.
Right. Now you got it. He teaches at Columbia University.
So pre-Columbian is before Columbus. Yeah, right. That's the ancient stuff.
So they have these little statues that they make to depict daily life.
And they have a Romagna. And
there was one with Romagna. I will dig it out of my
that's
files
pretty cool.
And put that in there too.
You have a picture?
I do.
All right. So what causes... I know we're jumping ahead, but...
We are.
Are there parasites in your eye?
Let's start with the how you catch this infection part.
So a reduvid bug bites your face.
Yeah, and then what happens?
It defecates and the feces have some form of the parasite, probably polymastigote.
Metacyclic trypomastigotes.
See, I did make many syllables. Metacyclic trypomastigotes.
That's correct.
Are these sexual parasites?
No. We don't, that's an interesting question, because it was debated for a long time as to whether or not trypanosomes are sexual or not. Mm-hmm. And I'm not sure that's ever been resolved, although some claim they have and some claim they haven't. If we typed that out on PubMed and asked the question in a different way, perhaps we can find out the answer.
All right, so basically you have these pro-apocypomaglagotes.
This is getting to be a lot like car talk.
It is getting to be. This has been from the
Schmendrick mastigote. And this is the good part of the new year. It gets worse, folks. It gets a lot worse. Okay.
Repeat the...
They're called metacyclic.
I should get the book, right?
You probably should.
In the early episodes, I used to have the book open.
You did, and sometimes you would quote it, and I would say, I wrote that? But yeah, sure we did.
Let me get it. Hold on.
Please do.
Fill the void.
Fill the void. Isn't it a prohibited void where prohibited?
No, without me, there's a huge void. Okay. What chapter is it?
It's early, because we did the trypanosomes. You've got it right in front of you. Right there. Bang. Ba-bum. That's the one. And there it is.
All right, kissing bug.
Yeah. Metacyclic trypomastigotes.
They're just called trypomastigotes here.
I know, but they're called metacyclic.
In fact, in this paper, trypomastigotes are going to play a role.
They are.
Right, that's part of their assay.
They are.
Trypomastogotes enter the wound.
Enter the wound, or
No, it could get in your eye too, right?
That's
so
right.
you could, in your sleep, you could rub your scratch and then rub your eye, and
Sure.
you would inoculate yourself.
That's correct. So the invasion process starts at a mucous membrane, perhaps.
Look, it's a chagoma.
A chagoma.
I'm sure we mentioned this.
We did. I know we did. And that's called Romagna sign because the doctor that first described it, his last name was Romagna.
Mm-hmm.
Okay. So that begins the infection. And if you know you have it at that stage, perhaps giving a drug would help.
So if you had someone come in with a sign of Chagas, which is a Romagna,
yeah, so that's unilateral.
how long after the bite?
I wanted to back up
sure.
present you with a choice. Because if you're a physician and someone just brings your kid in to see them, and you take a look and you see unilateral periorbital edema, you'd think of one thing. But what if they brought in a child that had bilateral periorbital edema? That's not Chagas disease at that point.
You think it could never be Chagas?
Well, if it's bilateral, then that's got to be something else.
Yeah, but there's always an exception to every biological rule.
It's true that I wouldn't doubt that in the least.
And in fact, why is it only one eye?
That's a good question, Vincent. Because when the bug bites,
it's
it
on
usually
one side.
defecates only on one side, and it induces itching.
On one side.
And the itching causes the child to scratch. And when that occurs, there's this fecal pellet, which is moist, containing the trypomastogotes. And those are the organisms that are rubbed in, and usually into one eye. By the time the kid gets around to rubbing the other eye, if they do, because you usually only sleep on one side, then the organisms have dried out and they're not infectious.
But in heavily infested areas, you could imagine a kid being bitten twice.
You could.
I bet it happens.
Romagna sign doesn't always happen. If it bites you somewhere else, and you should inadvertently put your fingers in your mouth, you can get infected that way too. How do I know that? Because
Chagoma is 2 to 4 days. Okay.
Because we talked about this with regards to a popular soft drink that's being sold on the beaches of Brazil that's made out of sugar cane.
Mm-hmm. We did it in TWiP?
We did. And then they press the sugar cane, but occasionally there's reduvid bugs in the sugar cane.
Oh, and they transmit infection?
And you can catch it by swallowing it.
Oh, interesting.
Very. So it's a mucus membrane. It's still another mucus membrane.
So you think it will invade in the mouth?
It will. You already know it will. And by the way, the same is true for the African trypanosomes as well.
Nice.
Except the African trypanosomes are injected by the tsetse flies. They're not rubbed into the bite wound. They're injected by the tsetse flies. But, pardon me, if you're a lion and you bring down, let's say, a gazelle or a wildebeest, and you begin gnawing away on these animals because that's your meal, and your teeth and your gums start to bleed because of the rough stuff that comes into your mouth, and the animal that you're eating happens to be infected with Trypanosoma brucei rhodesiense, let's say, then the lion can become infected by just eating their prey.
And would that be a brucei?
Oh, of course, sure.
African, right?
That's right. There's one over there called Trypanosoma evansi, which can be transmitted sexually in horses by sexual intercourse.
Trypanosomes actually are intracellular. No? They enter cells. What do you mean no?
Not the ones in Africa. The ones in Africa are
American.
Oh, you had to qualify it.
Yeah, because that's the
one. Because
you
said, well, trypanosomes are. You have to be more specific.
Okay. So I'm talking now about the American because I want to put it in the context of the paper.
Oh, but of course.
Which looked at T. cruzi.
Sure.
Is there another American species besides cruzi?
There are. I
think they mentioned one here.
There are several others.
They said here.
Yes, there are.
We should go to the back.
I'm blocking on their names right now.
I'm going to get it for you because they mentioned it and they said because of a lack of. They put it in this interesting way.
Right.
Because of a lack of. Fill the void, Dickson.
Sure. Trypanosoma vivax?
No.
Trypanosoma. I'm going to go down my list, but I do not recall which one is an animal parasite. It's not a human parasite. or a parasite of humans, I should say, that is also transmitted by reduvid bugs. In fact, all of the trypanosomes in the New World, considering this is the New World, are transmitted by reduvid bugs. And you're not finding the name of it, right?
Are you having problems?
I don't have any problem except that I don't remember the name. If that's a problem, then there's a lot of other problems that we can talk about too.
It's interesting.
Doesn't
maybe
come up?
No, it doesn't. Maybe when we scroll through the discussion, it'll come up. I remember them saying we have to check this strain, but we didn't have the facilities or the
PCR
resources,
Oh,
something
no,
like that.
Okay.
Anyway, so this paper, they're looking for...
So let's start with the infection. Let's start with the infection, right?
Oh, you want to start with the beginning?
But of course, because that's what this paper is about. How does this parasite gain entrance into cells? Because it's an obligate intracellular parasite.
The American ones.
Yeah.
It gets into cells.
It does.
It's not an apicomplexer, right?
No.
Good.
Is
it taken up by endosomes?
It's a hemoflagellate.
Hemoflagellate?
Yeah, that's what it is. It has a kinetoplast. It's a kinetoplastid.
Does it eat blood?
It's related. It doesn't eat. Well, let's see what. No, it doesn't eat blood. It's related to the Leishmania.
All right.
So the Leishmania and the trypanosomes are related to each other because they both contain kinetoplasts, which are extranuclear sources of DNA plus a cassette's worth of enzymes, which relates to its ability to process energy. So it's a remnant of a mitochondrion.
Okay.
Right? So it's got partial Krebs cycle capacity as well.
All right. So this T. cruzi needs to get inside of a cell.
That's correct.
How does it do that?
Right. And that's what this paper addresses. So it has a number of factors that it's
taken up by endocytosis, right?
Well,
you know what you say in your book?
Go on. You want to make me eat my own words here?
So if it attaches to the host cell.
Yeah.
Mediated through galectin-3.
Really?
Well, that's what you say. I don't know if it's true because this book is old.
It is. It's 2005.
You should revise it.
We're working on it.
But too slowly. We're going to have 400 TWiP episodes before you revise it.
That's the way it goes. That's just too bad. You can't rush quality.
Parasite can penetrate a variety of cells.
Right. In fact, it can penetrate almost all cells.
And then it enters a parasitophorous vacuole.
That is right.
It enlists several escape mechanisms to aid its survival. It neutralizes the pH.
One.
Produces a number of proteins which allow it to survive. Chagasin is one, a cysteine protease inhibitor.
Yes.
Which
but
is
it
necessary.
Also secretes a cysteine
Listen to
protease.
this. Coming up, it actually needs to inhibit lysosomal proteases so it won't be degraded, but it does make cruzipain.
That's right.
And it says cruzipain is thought to play a major role in helping the parasite avoid being digested. It's the same thing.
Right. Well, it's an anti-enzyme which digests enzymes. All right.
And it's also believed to be involved in inhibiting apoptosis.
Right. It's a cysteine protease. It's
cruzipain.
Cruzipain. That's
a cysteine protease.
That is correct.
And that means in the active site, one of the residues is a cysteine.
This is all correct.
Right? Which is an amino acid.
That's right.
Or an amino acid.
It's related to the cathepsins, and cathepsins are all lysosomal-based proteases.
Did you know that cathepsins play a role in...
viral infections.
Yes, but in a normal human...
Yeah, well, of course they do. Otherwise, we couldn't digest
bone reabsorption. In fact, there is a cathepsin inhibitor in phase 3 for postmenopausal. What's the bone disease caused?
Osteoporosis.
Thank you.
That's great. What about astronauts that spend time in outer space?
Their bones get soft.
There's a calcium uptake flux which exceeds the bone laying down flux.
Is that a good reason to stay out of outer space?
Tell me about it.
And give R1s instead?
There you go.
With that money?
Oh, yeah.
Oops, sorry, I didn't say.
I would love it. So,
I do understand. I'm sorry about that spring noise. I'm moving my mic around.
That's right.
I'm a big fan of science.
Right.
But I'm a bigger fan of microbiology.
No, that's not different, actually.
Which I include parasitology.
Oh, thank you.
So we have to get into a cell, and we make these cruzipains.
We do.
And in this paper, they actually say these are essential for the viability, infectivity, and virulence
How do they know that?
T. cruzi.
And how do they know that?
I would guess that inhibitors have
Roger
been
that.
made
Roger that.
that interfere, right?
Roger that. Or you could do a knockout.
Can you?
Yeah, you can actually knock out these things.
Dickson, there are a couple of drugs available for treating.
There are.
Do they?
They're awful.
They don't hit the protease.
No,
right?
they don't. One's called benznidazole, which is a horrible drug, and the other one's called
Horrible name. Who would put a Z next to an N?
Nobody, but they did because they had to. That's reminiscent of the chemical
What's
structure.
the other one?
It's called nifurtimox. And there are other drugs too. There's called a sham therapy. That's for African trypanosomes, not for.
It's when you use a pillow.
No, it's a sham. It's a shunt.
You didn't get that, did you?
I got it. I'm trying to avoid it.
What? This is a New Year's resolution?
No, probably.
No more card talk?
No, we can do card talk if you'd like. So, in the case of the African trypanosomes, they use a penta shunt to derive most of their energy from because they're mostly anaerobic. Mm-hmm. And so they've derived a therapy. Alan Clarkson and his group at NYU and other people too have derived a therapy called sham, which interferes with that pentose shunt, which we don't make use of mostly.
Okay.
And so therefore you can knock out the organism this way too. But indeed, it's the toxic aspects of both benznidazole and nifurtimox are so horrible that it's almost better to not treat and just to live with this disease because it's a chronic long-term infection.
It's horrible.
It is. The organism starts out as a triple mastigote, and it enters, a metacyclic triple mastigote, enters the fluid portion of a mucous membrane. Mm-hmm. And then the magic happens. Then this organism somehow breaches the cell membrane, gets incorporated into a unique parasitophorous vacuole, throws away its flagellum, rounds up into a leishmania-like organism, and then it escapes into the naked cytoplasm. It lives in naked cytoplasm.
When does it reproduce?
It starts to divide after that happens.
So after it gets out of the cell?
Yeah, it's an escape
what's the
mechanism.
Purpose of
it's a Houdini.
What's the purpose of going in the cell in the first place?
It's a safe haven. If you can avoid the innate immune response, it's a safe haven. And that's a big if, right?
Well, you have to avoid the lysosomal things as
that's
well.
That's number one.
But so getting in the cell is not a free ride.
No. And you have to avoid all those immune responses that are associated with toll-like receptors, right? Right. So this deals with that. This parasite has to deal with that as well. So there are how many toll-like receptors?
11, 12.
11 or 12. And what mice have a couple of that we don't have. Is that true? Mm-hmm. They have number 10. We don't have number 10. In this case, toll receptor number 2 is the one they talk about mostly, as I recall. And toll-like receptor number 2 and IL-6, as I recall also from the paper, are linked together to elicit an immune response which has no effect on the parasite. So it's a diversionary tactic. And it allows the parasite to escape from the parasitophorous vacuole into the naked cytoplasm. So I call it hiding in plain sight. It's living inside your own cytoplasm. It's like it was another subcellular particle, like a peroxisome or a mitochondrion or a centriole. This parasite lives the life of an intracellular particle, and yet it replicates. So it derives its livelihood from nutrition that it obtains from the naked cytoplasm.
All right.
And we're really not sure of how that works. But maybe this cysteine protease plays a role there too, because cathepsins obviously allow organisms, including us, to digest proteins and to recycle the amino acids back into their own proteins.
Well, this cruzipain does a lot of things, apparently.
It does a lot of
Wouldn't be
things.
surprised if it did even more. It says here, I found a review article. It's involved in multiple functions, cell invasion, host tissue proteolytic degradation, evasion of immune responses.
Does that mean that they haven't really pinpointed what it really does yet, and they've got these as epi effects, these side effects perhaps? Or is it real that it does all these multiple things?
I think probably some. I mean, I'm just reading this abstract where they make very broad statements, so I would bet that there are some specific mechanisms known.
Right.
So it's a good target because it's involved in so many things.
And it's in all the stages, by the way. It's in all the triple mastigote stages inside the gut tract of the reduvid bug. It's in the triple mastigotes that they defecate, and it's in the amastigote stages inside the cells. So it's a universally present enzyme.
Okay.
So it's a target. It's a great target.
By the way, I had a question. They mentioned in this paper, besides vectored transmission, there's mother to child.
There is.
Now, how does that work?
Well, let's say, and by the way, keep in mind that this parasite doesn't discriminate with regards to cell type. So although there's a penchant for it to accumulate in the myenteric plexus in the gut tract, which is nervous tissue, it can also affect cardiac tissue. It can also affect macrophages. So this organism has the ability to infect a macrophage. And if macrophages can cross the placental barrier with the parasite inside, as we think happens also in an apicomplexan parasite called Toxoplasma gondii, then that's how the fetus could become infected.
I see. So they say here that the non-vectored transmission is becoming an increasing
It
public
is.
health threat.
And by the way, in the United States, just to relate this to a local thing, there are many people living in the United States now from other places. And the trypanosome that we're talking about is found from the Texas-Mexico border all the way down into the cone region of South America, meaning Chile and Argentina. And a lot of those people unwittingly have brought their non-clinical phased Chagas disease with them. And so they have given blood. And some of them have donated organs, too, when they died. And those are transmitted to other people through transfusion, trypanosomiasis. Now, there's a very famous couple, Victor and Ruth Nussensweig.
Who we have mentioned before.
We have. And Victor became famous. They're both from Brazil, by the way. And the one thing he did for Brazil was to invent a treatment for blood using gentian violet, 1 in 4,000, which if you treat the blood with gentian violet and then transfuse a patient with it, it kills off the trypomastigotes in the bloodstream and you don't get Chagas disease.
Cool.
Now, we could do that here, but we don't. And probably because we consider it too trivial. But I bet you if you looked up on CDC's morbidity mortality reports for the number of cases that have been transmitted from transfusion to patient, you'd find it's in the hundreds, maybe even the thousands up to now. By the way, this results in a cardiac condition called megaesophagus, megacardia. Okay, it's the single leading cause of cardiomyopathy in the world. It's estimated there are over 12 million people that harbor this parasite, and a lot of them suffer from the heart disease aspect of Chagas. So it's a huge problem in some areas. And so it's worthy of attention from the chemotherapeutic side of things. So here's this paper. Now we can discuss the paper.
Cruzipain.
Cruzipain is the target.
And they have previously shown that you can make inhibitors, chemical inhibitors of this, and they will inhibit the parasite.
Correct.
So they wanted to pursue
and they're
that
reversible, right? Because it has to be reversible. Otherwise, it would inhibit our own cysteine proteases, right?
Yeah, so that's something that we'll get into, that the cathepsins are related to cruzipain, and you have to have some selectivity, otherwise the drug will be toxic, right?
Exactly right.
Now, these inhibitors, the previous ones, so they say they've been just discovered recently, reversible cathepsin K inhibitors. Now, these are inhibitors of cathepsins.
Right.
They have a nitrile warhead.
Oh,
isn't that great? I mean, I think that's a title, don't you think?
Almost like a Nike warhead.
A nitrile warhead.
Yeah. So I have a question here, Vince. Do you
know what a nitrile is?
I do.
Okay. What is it?
It's a group that has a specific chemical configuration reminiscent of a nitrile group.
All right,
that's
so you don't know.
1960s singing group that likes to sing in the men's room.
You don't know what a nitrile is.
You'll tell us.
C triple bond N.
It's got an acetyl bond.
No, it's not an acetyl.
It's a hetero.
It's a carbon.
It's a hetero organic triple bonded compound.
Heteroorganic. Come on. It's a carbon with 3 bonds
that bonds to
connected
nitrogen.
Yeah,
Right.
triple bond.
That's incredible.
That is a nitrile group.
That's incredible.
There's another name for it.
Yeah.
It's also called cyano,
Okay.
a cyano group.
Because I remember acetyl groups were 2 carbons with a triple bond. Is that correct?
No,
Acetyl.
acetyl is totally different.
What would you call a triple bonded carbon-carbon compound? Let's look it up. That takes us back to our old organic chemistry days, which are failing us miserably right now, by the way. That's part of the car talk stuff. Yeah, what's the triple bond called?
Between
2 carbons.
Oh, I see.
Yeah, it's got a name, like a nitrile group.
Alkyne.
Alkane.
Alkyne.
Alkyne. Okay.
Any alkynes have
Okay.
carbon-carbon triple bonds.
There you go. Okay, so nitrile has a triple bond and alkyne.
Nitrile,
Is
also
there-
known as cyano, and nitriles are useful for many different things.
Well, cyano dyes. They use them a lot in dyes, don't
Yeah, so you know what else that's in crazy glue?
they? Really?
Crazy glue contains methyl cyanoacrylate. Cyano means there's a nitrile group in there.
I got it.
And now these inhibitors, these reversible cathepsin inhibitors, also have nitrile groups. So, in fact, they have pictures of the compounds in the paper. They're huge.
they do. They
They're
do.
huge chicken wire, dihydroxy chicken wire,
Right.
and then there's one nitrile
One
group.
big active group with the R. That's right. That's the active
Right,
group.
well, just one.
I have a question for you,
Yeah.
Vince. Could you do the audience a favor by describing the definition of therapeutic index?
Why?
Come on, your wife worked at Merck. She must have come
Actually,
every night talking about this.
this
This
paper
paper
was-
deals with that issue.
How much you need to inhibit?
No, but what is the definition of a therapeutic index?
Therapeutic,
Index.
the amount of drug you need to inhibit 50%.
No.
The comparison of the amount of a therapeutic agent that causes the effect to the amount that causes death
Right.
or toxicity.
To the patient. So when you do drug trials and you want to know whether or not you can kill the organism without killing the host,
Yeah.
you have to calculate a therapeutic index, right? And the bigger the therapeutic index, the more of the drug you can apply to the problem. And the lower the therapeutic index, the more dangerous that drug is. And eventually, you might end up with, if you give X amount of drug, you kill the organism. If you give X plus 0.001 of the drug, you kill the patient.
Well, when in human trials, it's not going to be killing; it's going to be toxicity.
One, that's the
Right.
therapeutic index of one.
Or
means
side effects,
Forget
yeah.
about it. You have like 1 in 2,000
no
or 1 in
good.
20,000. There was a famous drug trial with a drug which we now call puromycin, which is not a drug. Right.
Puromycin is an antibiotic, right?
Well, puromycin is a protein inhibitor,
A
right?
protein synthesis inhibitor.
right?
Yeah.
And the American cyanamide company up in the Pearl River decided to treat, guess what? Trypanosoma evansi. Trypanosoma evansi. That's the sexually transmitted horse trypanosome.
Okay.
And they wanted to try it out on horses because if they had tried it on people, that would have been bad. So the first day of their drug trial, using puromycin as the drug of choice, it completely wiped out the infection.
And
in
the horses too?
Well, that was the next day. When they came the next day, all the horses were dead. So the therapeutic index for that drug was listed as 1.
You know, we have listeners who like horses,
I know.
and you're laughing that they're dead.
No, but I'm laughing that they thought they had a great drug, and the next day they thought they had a horrible drug. They didn't have a drug, right? Because it killed off the horse the same way it killed off the parasite. So the index was 1. That's where they got the drug therapeutic index from. So from that point on, all the other drugs were measured in terms of, if you give 1 dose and kills the parasite, how many doses of the same drug do you have to give to kill the host? And that's why the
Right.
drug trials are based on that concept.
Therapeutic index.
Therapeutic index. And all the physicians talk about it all
There are a
the
few other terms that we're going to come upon here,
time. I hope so.
like pharmacokinetics,
That's good.
and there's also another one, pharmacodynamics,
Pharmacodynamics.
bioavailability.
Bio and degradation.
Is all this stuff is what
Yeah,
they
drug stuff.
That's what my wife does. Yeah,
Drug stuff.
pharmacology of drugs.
And here's a parasite, and she's doing a cardiotoxic or cardiodetoxic drug right now for a company.
Yeah.
This is a cardiotoxic organism.
Well, she was probably aware of this program.
Well, she
when
did
she
a-
first went to Merck, she worked on
Yes.
ivermectin,
Right.
which inhibits,
Right.
it's used to treat river
Yes,
blindness, right?
That's true.
Which
A
is
worm.
caused by,
It interrupts the nervous system of the worm.
what's the worm called?
Onchocerca
Onchocerca
But didn't you work with
volvulus.
George Cross also on trypanosomes?
Yeah, at Rockefeller.
Aha.
Trypanosoma brucei, probably.
Right.
That's what they work with
so
in
that
the
was
lab.
a different one than this one. Yeah. No one in their right mind in those days would work on T. cruzi. You know why?
No.
Because if you caught it, there was no cure.
If you caught brucei, you would take this NZ drug.
Well, no, you took another. That's not the drug they were using. They were using. Oh dear, I'll try to remember the name of that drug. There was another drug that they were using for African trypanosomiasis that was probably effective.
Well, these guys used cruzi, though.
They did. No, they were working on this.
Because we have two drugs now that
That's
you can
right.
use if you get infected.
That's right. So let's talk about the paper.
All right, so if you were going to do this,
Yes.
and you knew that for cathepsins you could make drugs, nitrile compounds that inhibit them, how would you find nitrile compounds that inhibit cruzipain? How would you, Dickson Despommier, do that?
Well, now that we have the complete genome of Trypanosoma cruzi,
Yeah.
I can make a construct for cruzipain. For cruzipain. I can get the gene for cruzipain.
You can express the
I can
protein.
express the protein.
Okay.
And now I can deal with the issue of just the cruzipain and nothing else.
Okay. So now you can say, here we have the enzyme, let's inhibit it.
Right.
So how would you identify inhibitors?
So what I would do then, because I now can bring this up to date in terms of modern chemistry, right? They now have a way of generating a three-dimensional structure of the enzyme. And they have a way of envisioning the pocket that you want to inhibit. And then they have this enormous library of compounds that you can ask the computer to compare to the size and the shape of the pocket and the charge. And eventually it will select out from a huge list of potential compounds, likely compounds that will combine with the active site. That's what I would do next. I would do a literature search and a computer search for compounds that had a reasonable chance of interacting with the pocket. I would then, A, have them synthesized, and B, I would do test tube, measurements to see whether or not you would inhibit the cruzipain by using a protein that's a cruzipain axon.
So that's what they did. They had a 900 compound library.
A library, the peptide library.
Of nitrile-containing inhibitors.
Yeah.
And they use the computer to say which one of these are likely
Why
to
did they
fit.
pick nitrile, by the way, to begin with?
Because there are nitrile inhibitors of cathepsins, which are in phase 3 for postmenopausal osteoporosis.
But there are other diseases of lysosomes also that you would love to be able to inhibit using anti-cathepsin agents, right? And I think one of them is called arthritis. Yeah.
So they identified a few, and then they study these. Actually, they identify a few, and they study these in this paper.
So they found the ones that had the highest rate of inhibition?
Well, they say we identified in this library a few that looked good, and then we optimized them for potency and pharmacokinetic properties. They had the chemists mess with the play with the side chains, and then they got 3 inhibitors: weak, moderate, and potent inhibitors of the enzyme, as you said, expressing the enzyme in vitro.
So what's the biggest problem of, even if you identified a perfect compound, what's the biggest problem you'd face in treating a patient with Chagas disease with that drug?
Well, there are many problems.
Well, cost is one of them. Let's eliminate
but no,
that
even before, assuming you get a drug licensed, is that what you're telling me?
No. Just to get it to
because
where
first of all, it has to be bioavailable. So if you feed
What do
it...
you mean by that?
Well, if you feed it... So it's nice to be able to give a pill, right?
Oral.
It has to get to the place where it needs to be active. In this case, I presume the blood.
So
that bioavailable means...
not in the blood. No, it's not active in the blood. That's what I'm raising.
Where
the point
is
about.
the parasite?
It's intracellular.
So you have to go in there. Well, some of the assays don't look intracellular phases. They're looking at extracellular.
I know that because that's the way the organism travels from tissue to tissue. It redifferentiates into a tripomastogote, gets into the bloodstream, and then travels to another part in the body and then infects some more cells that way. But really what you want to do is you want to knock out the parasites inside the cells that are damaging heart muscle, megacolon, all those tissues that
No,
are
in
being
fact,
damaged.
what they do in this paper is
I understand.
they feed the mice,
Yep.
and then they take various tissues, including blood,
Wait.
and ask, does the drug get there?
Now you just mentioned something else that's
To
interesting.
Dickson, sometimes for some of these compounds, it never gets to a tissue from being fed,
So
right?
that's one.
So that's what bioavailability is.
But they don't have to feed it necessarily. They can inject it.
In this study, they put it in their chow.
I understand that. But what was the big advantage of using T. cruzi? Do you know, we didn't discuss this yet, but we will now.
Big
Where-
advantage?
Yeah, there's
Is there a
huge-
mouse model?
It's not a model.
It's a mouse pathogen?
Yeah.
It's naturally a mouse pathogen.
Yeah. What else is it a pathogen of?
Well, humans, right? T.
What else is
cruzi.
it a pathogen of?
Cows? Cows?
What else is it a pathogen
Monkeys?
of? What else?
Everything.
Yes. This is
Affects
a
everything?
non-discriminate parasite.
It's like
If you-
toxo, huh?
Yeah, a little bit. If you go down to the South American jungles of Brazil and you find a mammal, it's likely that that mammal will be infected.
So every animal is a model.
It's
so
not a model.
T. brucei is different?
Much different.
Which animals are
So
infected?
in East Africa, all of the game animals are infected.
Yeah,
In West Africa, only pigs and people.
interesting.
Very interesting.
Is that because the parasite is different or the animals are different?
No, we know that the parasites are different because there's a barrier, right? It doesn't go any further over,
Yeah.
and it doesn't go any further the other way either. That's very interesting. So that's probably vector controlled rather than parasite controlled. The point is that these guys are working on a parasite that no matter which animal species you try this out in, it's going to work.
Okay.
That's very fortunate because if you start with mice, that's just the first screen of many, right? You really want to work up through primates and then eventually
Yeah,
with
this
people.
paper is a mouse, but it's a preliminary study.
Exactly.
as
What
you
if it
know.
does absorb in a mouse? Does that mean it will absorb
No.
in a monkey? Of course it doesn't.
Absolutely not.
That's exactly right. So you'd have to do this
Ferrets are
again.
not
and again.
humans.
We're trying to ferret out the data here. Come on.
So the first set of experiments, they take these hits and they do your assay. They make the enzyme and test it in vitro.
Let's say it's perfect. The mouse is totally cured, 2 doses. They never even get sick from it. And the next thing you know, they're off and running again. And they're perfect.
So
Then
they
what do you do
have
next?
wait, before I just want to tell you
No, okay.
some of them have nanomolar inhibitory levels. So 1 to 2 nanomolar. That's pretty good, right?
It's darn good.
Then they have one that's like 170 nanomolar, which is moderate.
Yeah.
And then they have a 4 micromolar, which is weak. So for a drug, you don't want
The
micromolar,
higher the
right?
dose of drug you give, the more likely it is that it has a low therapeutic index.
Mm-hmm.
So
All right.
what's the big advantage of ivermectin? You mentioned that drug before.
It has a very high therapeutic
Not
index.
just high, it's an enormously high thing. And not only that,
It's not toxic at all,
you
is
know,
it?
well, it could be if it gets across the blood-brain barrier, but it doesn't because it's huge. It's a dihydroxy chicken wire compound. And that means that it won't cross the blood-brain barrier. And the other thing is it's long-lasting. You give one drug a year, a dose of drug a year.
For
and it's-
ivermectin?
For ivermectin, and it's curing the infection for 1 whole year for onchocerciasis.
So in the business, they would say the PK and the PD are very promising. The
They would say that.
pharmacokinetics
They would say that.
and the pharmacodynamics.
Right. And if it's low doses that you're giving, it's cheap to make too, especially
Yes.
with ivermectin because that's a fermentation
And,
product.
and that's why Merck gave it away, right?
Well, they didn't want to.
It was so cheap and it was good PR.
Bill Bradley sort of pushed them into doing that.
The basketball player
Yeah.
and Rhodes Scholar,
And
senator from New Jersey.
That's correct.
Is he still around?
Oh, sure.
He-
Just in a belly full of politics. He just got out. He just couldn't stand it anymore.
what is he doing?
I think he's running a law office.
Come closer to the mic. You
He's
can't.
running a law office.
You can't sit back and relax. If you
I'm
wanna
not
do that-
relaxed.
if you want to do that, you have to wear a headset.
I'm excited about this. I'm really excited
So
about
after they did these enzyme studies in vitro,
this. Yes.
they moved into parasites. So
That's
let me
right.
ask you a question about these
You
assays.
please do that.
The first assay is called an epimastigote assay. What is an epimastigote?
Epimastigotes are the ones that are found inside the gut tract of the reduvid bug. That
And
you
that's
can
the one that you
culture.
get. That's the
You
one
can
that you
culture
get.
them.
Meaning culture, you put them in some medium and they will reproduce.
That's correct.
So you can't move away from the mic.
What is
Dixon.
this called here?
A tropomastigote.
Trypomastigote, right? So let me show you another
Tripo.
picture. I'll show you another picture.
There's a epi. When is it no longer an epimastigote?
when it comes at the back end of a reduvid bug. Oh, I don't have a picture
Oh,
of
so inside the bug, it's an epimastigote.
it. Yeah, no, I think I illustrated
Why would
that.
they test an epimastigote? You're never going to see drug in a reduvid bug, right?
What if, because it's the stage that you can culture.
You know how they measured inhibition?
Tell me.
Incorporation of tritiated thymidine.
Right.
What is that
Just
measuring, Dickson?
division of DNA.
Yeah, DNA synthesis.
Yeah, well, there are two sets of DNA synthesis in that organism. One is in the kinetoplast, and the other is in the nucleus.
So they could show that adding these drugs,
Yeah.
cathepsin, not cathepsin,
No.
cruzipain
Cysteine
inhibitors.
protease. That's right.
These inhibitors, they identified they could inhibit DNA synthesis of epimastigotes in
But
culture.
it tells you that it's not just essential for invasion; it's essential for reproduction too.
Right.
That's interesting. So
But listen to this, Dickson.
I'm listening to this, Vincent.
There was a 1,000-fold difference in the potency versus the enzyme versus the epimastigote.
It takes a lot more to push it inside.
Are you surprised?
No, not really, because there's something called the flagellar pocket. The flagellar pocket. Now, the flagellar pocket on a
Is that a flagellum in your pocket?
No, I'm just glad to see you.
Isn't that funny?
But on bump. Yeah, well, I got the joke. So if you're an African trypanosome and you live in the blood supply, what do you eat?
Heme?
Silly. That's so silly. No, you don't eat. That's so
Globin?
silly. That is also...
Albumin.
You can eat proteins of various sorts. And
Well,
you can
albumin
absorb
is a
amino
protein.
acids across your membrane, but there's only one way to take them up. Pinocytosis.
It happens at
And
the
there's a
flagellum,
little
flagellar pocket.
Right
yeah.
into the flagellar pocket. That's where their food gets taken up. It's the only place that you don't have the variant antigen protein code.
Mm-hmm.
It stops right there, okay? So that's why people were looking at that as a target for antibodies, to try to knock out the flagellar pocket proteins and prevent the organism from feeding. Now, I'm going to assume that the trypanosomes in Africa and the trypanosomes in South America feed similarly, at least while they're in the blood supply. So the epimastigotes, although they're not in the blood supply, they can be cultured. So therefore, they have a flagellar pocket as well. And so maybe that's where these proteins are being taken up by pinocytosis. But you'll need more drug to get them into the organism that way than you would just working with the cruzipain outside the organism itself.
Now, in one experiment with these epimastigotes, they say the treatment resulted in the epimastigotes being elongated and slow-moving. This suggests that the drug had completely killed the parasites. Would you agree with that suggestion?
Well, if they were slow-moving, they're still alive. I'm sorry, but that's not dead.
They would like them to be dead.
They would love them to be dead. But they probably injured them beyond their ability to infect, although they could have tested that.
Right. Now, but they didn't. That's a good point. They did an amastigote assay. What's an amastigote?
The amastigote, well, what does anything mean?
Not.
Right. So what is the word
not.
Yes.
Not. No.
Who's a nut?
Who's on first?
Yes.
No, he's on third.
I don't know. That's
Second.
four base. All right. We don't even know the routine, but we still think it's funny. So
I bet we could do a good version of it.
we
though.
might if we had it up on the screen.
But we're not comedians.
I hope not.
No.
We're trying to be humorous in our exchanges, but we're not trying to be comedians.
Actually,
here.
I should just cut it out because it's silly.
No, it's fine.
I
It's
can
fine.
just
I
hear
don't
our
mind
listeners.
it at all. I don't mind it at all.
Well,
Vincent.
what you mind doesn't matter.
This is all true.
It's all in the numbers.
True.
Just if people stop
Right.
listening, then we won't be funny.
That's right. So
Amastigote.
an amastigote
It's
as
lost
opposed
something.
to a mastigote.
So it's lost something.
So what does the word mastigote refer to?
Masti means chewing.
Stop it.
Masticating.
There's an entire order of protozoans with this name.
What does it mean?
Mastigophora.
What does that mean?
It has a flagellum.
Yeah, so amastigotes don't have a flagellum.
So name some mastigote parasites.
With flagella?
Yeah.
Trypanosomes.
That's one.
Giardia.
That's good. Name another one.
Wait a minute. Trichomonas.
Very good. Oh,
Yeah.
that's great. I've always remembered that there are 7 ways for viruses to
So I have to
replicate.
remember which ones have flagella.
This one you have to remember, because I used to quiz our students like that to get them to think about groups of organisms together.
Are you going to ask me to name some without flagella?
No, because... Yeah, well, okay, name one.
Plasmodium. Do they have flagella?
No, of course not.
Plasmodium doesn't.
No, they're apicomplexa.
The cestodes, the trematodes, the
We're talking about
nematodes.
protozoans now. Protozoans.
What?
Protozoans. Single-cell organisms.
Oh, you want me to name you a single-cell organism?
Other groups.
Cryptococcus
Yeah. That's an apicomplexa, right?
doesn't have a flagellum.
No, of course not. It's not a mastigophora.
Okay. So I got it. So an amastigote has lost the...
It has. So here's an organism that can be both.
Where do you find an trypomastigote?
No, it's not called a tripomastigote anymore. It's called an amastigote.
Where do you find it?
You find it intracellular.
Yeah, you had said before that it loses the
So when it
flagellum,
penetrates the cell,
Okay.
or it gets taken up by the cell in a certain way, if Howard Schumann was here, he would probably say, oh, and I have an organism that also gets taken up in a strange way, too, because he works on Legionella, right? Mm-hmm. And the Legionella induces a very interesting corkscrew-shaped parasitophorous vacuole, which the organism can then live in without being digested by the lysosomes. This organism takes it one step further, goes into a parasitophorous vacuole that's induced by the parasite. The host responds by doing it. The parasite then secretes a pH-adjusting compound, which neutralizes the lysosomal enzymes, then secretes out the cruzipain, which digests up the lysosomal enzymes so it no longer has to worry about adjusting the pH. And then, to add insult to injury, it drops its flagellum as it penetrates beyond the parasitophorous vacuole and enters the naked cytoplasm of the cell.
All right, so in this assay, they look inside cells and they count the number of amastigotes.
Tougher to get a compound through the host cell,
Right.
then into the naked cytoplasm, then into the parasite.
So they do
three
with and
things to
without
worry
drug.
about. With and without
And
drug.
they also use a known inhibitor, which would be K777.
Right, which is... It's awful if that's how many compounds they had to try before they hit a successful
Let
one.
me tell you what K77... I should have told you this before because they used it previously.
You're telling...
It's a basic cathepsin inhibitor.
Oh, okay, fine.
Basic meanings it will target the lysosome.
Got
okay.
it. Got it.
And these all...
For treating osteoporosis.
Exactly.
Okay.
Basically, these all inhibit
Protein.
amastigotes.
Right. But now what's
Okay.
the therapeutic index here?
Hang on, one more experiment.
Okay.
Next, they look at trypomastigotes.
Yep.
Trypomastigotes released into the medium, they call
Correct. Now, remember I told you, in order for the parasite to go from tissue to tissue, it has to revert back to the trypomastigote, the one that got into the cell to begin with. It has to convert back to that.
it. Okay.
And the flagellum grows out of the kinetoplast. Okay? So that generates the
All right.
kinetoplast.
So they say they count the number of trypomastigotes in the medium that have been released from these infected cells.
Right.
And all these, there are new drugs in K77. They all better than 95% reduction in.
There's something a little bit wrong with their assay, though, I would say, because how would they know whether or not the drug prohibited the formation of a trypomastigote or it just killed them off?
Well, they don't know, right?
Right.
They're just looking at release
I understand.
because they would like to know as many different steps
I understand.
as possible, right?
Got it. Sure. You wanna
Okay,
go
now we move to mice.
Aha.
In this
That's
section,
from the in vitro, from the isolated enzyme to the in vitro systems to the in vivo systems.
they call this in vivo dosing paradigms.
Love it. Love
So
it.
they first started by giving them the drug in the drinking water.
Right.
So
Sound
that-
familiar? That's how ivermectin was given
To
to mice that had parasites
Right.
of various sorts. And that's how they found it.
and they adjusted the concentration
Right.
so that they would get 50 milligrams per kilogram.
Assuming that the drinking rate is the same for the mice, but that's a little...
One of the three compounds had a bioavailability of 4%, which meant that from the water to whatever tissue they looked at, only 4% of the compound got there. So
But was it enough?
no, they didn't do anything more with that. They said that's enough.
How do they know that it was not enough, though?
They said it's unsuitable for in vivo. 4%, they wouldn't even bother.
But what if only picogram amounts are necessary?
Well, we know already that you need at least nanogram amounts.
Well, depends on how much you put in the water.
Well, listen, Dickson, they say here it was unsuitable making it unsuitable for in vivo.
You know what? Okay.
You could argue with them.
I'm not going to argue with them. I'm just raising it as a question. That's all.
I think it's a good question, but I assume that if it were picogram availability, sure, but it's not. It's nanogram.
But wouldn't you want to try it on an infected mouse anyway, just to make sure?
I would.
What's
but
the risk?
Money. It costs money.
Come on, 5 mice.
You got to pay the people who do the experiments.
Stop it.
Got to pay the electric bill, the water bill, the fuel bill, the internet bill.
In terms of the payoff, what if it worked?
Yeah, but you have to listen. In the industry, you have to consider what experiment to do. You could do lots of experiments, but it's very expensive. You have to be smart. The best people in industry are the ones that know which experiments to do, because you can't do them all. Like here in the academics, we can do them all.
Do you think they miss a lot of results as a result of that?
I'm sure.
Yeah,
I'm sure they do.
Me too. Okay,
so for example, another drug, one of the other three had a 45% bioavailability, so they picked that one.
That's pretty good.
And
the mice didn't die?
Well, wait, some of them do die. But the problem was they had suboptimal pharmacokinetic properties. Because these drugs have short half-lives.
Right. So they're biodegraded.
They're biodegraded, basically. So they say this is no good.
They don't get to the active site in time, and therefore the animal is not cured.
Here's part of the problem. So even when they pushed it into their stomach, pharmacokinetics are no good. By gavage, you put a tube in. So then they decided to put it in the chow, because they say mice eat all day long.
They do.
And in fact, that was better. They formulated it into the chow, and now it's nicely bioavailable, and the pharmacokinetics are better. It's more stable when you put it in the chow, mostly because they're eating it continuously.
What about just injecting it intraperitoneally into a saline solution?
Yeah, I guess, but they didn't do that here.
Right.
I don't know why.
Okay. I mean, I would have thought of doing that too.
So they got good tissue levels when they put it in the chow.
Okay.
So now they can go and do the real experiment.
Okay.
So far they spent a year figuring out how to deliver enough to the right places in mice.
Right.
It's not easy, Dickson.
No.
You don't just make a drug and give it and boom.
I never said it was.
Well, some people may think it's easy, but
it's never easy. Nothing
worth work.
Doing is easy.
So they inoculate mice with T. cruzi. Yep. What forms would they inoculate them with?
The epimastigotes.
And how would you inoculate them? Inject them? You want me to look it up?
Could.
Want me to look it up?
Sure. You could inject them intraperitoneally.
I thought you would know, but I guess you just don't know.
Intraperitoneally would be just fine.
That was a little dig, I'm sorry.
That's quite right.
Okay, in vivo murine efficacy studies. Are you ready?
I'm ready.
Of course, they had to grow the strains up.
Bring it on.
They made tripomastigotes for injection. Those are the released forms from the cell, right? And they actually...
the epimastigotes will transform into tripomastigotes.
They purified them on a column.
Really?
Yeah.
Interesting.
Yeah. And how did they put it in the cage?
Right.
I'm sorry. How did they inject the mice? What would you guess?
How did they inject them, or did they inject them?
No, how did they?
Well, I'm going to guess intraperitoneal.
Yeah, they did. And how many?
How many tripomastigotes? 100.
10,000.
That's a lot. You wouldn't need that many.
And they say a good number develop
sure.
parasitemia.
Yes.
So they actually check the blood
right.
as a quick way of seeing parasitemia, right?
Yeah, that's one way.
For parasitemia assessments, 10 microliter blood samples were taken from the tail vein, and then they just count it.
Right.
You look at it under a microscope.
Yeah, but rapidly these organisms become intracellular and amastigotes, and they become amastigotes.
And they use a Neubauer chamber. Do you know what that is?
Yes, I do.
Did you use one?
I didn't. I worked on worms, remember?
So you don't need a Neubauer chamber?
A Neubauer. We don't need your stinking Neubauer chamber.
Anyway, so they find that these drugs do reduce parasitemia.
Right. Okay.
Comparable to benznidazole.
Comparable.
Here they use benznidazole as a comparator.
Right. Very toxic drug.
Right. and there is no recrudescence of parasitemia
Meaning?
after stopping the dose.
Meaning?
It doesn't come back.
Gotcha.
Recrudescence.
Yeah.
Right, and they actually also used PCR to check.
So they got a sterile mouse in terms of the infection by a single dose?
Some of the mice.
That's amazing. You mean some of the mice? Some of the mice in the same group, or some of the mice getting a different dose?
Each group had at least some positive mice always. So, for example, the benzonidazole-treated mice,
yeah, it's not 100%.
after 90 days, 5 of 9 were parasite positive still,
That's not good.
50%.
That's not good.
And in one of their drugs, 1 in 10 was parasite
That's
positive.
not bad.
Yeah, but you don't want any, right?
I would like 0, of course.
But this is just a preliminary study.
Okay, fine. All the mice lived. That was a good thing. They didn't die of the drug.
No. Some of the mice died. In one study, 3 mice died, 1 in each of the untreated, 1 in benzonidazole treated, and 1 in one of their groups.
Right.
So,
but they'd have to do that for the controls, though. I mean, you have to have a control that's not infected that received the same amount of drug.
They died. So the untreated, some of the untreated mice died.
That's what I'm saying. They were.
So the
drug itself was toxic,
and the infection was also toxic, right?
Well, no, you have 3 groups.
Look, 3 mice died, 1 in each of the untreated.
One group you don't treat, and you infect.
One mouse died.
That's all?
Well, it's only 10 mice.
Of the all infected mice, only 1 died that weren't treated? They should have all died.
I think they arranged it so they wouldn't die, so they could have long-term follow-up.
See, that's okay. So
1 mouse in the untreated infected group died on day 63.
Okay. So it's a chronic infection, even in mice, then.
So these drugs have some efficacy in mice.
Right.
Right? So there's a reduction in
right.
parasitemia.
Right.
Nothing
is black and white. Nothing.
Of course. And they also did another experiment where so they would treat the mice or not treat them, infect.
Right.
And then after 90 days, they would immunosuppress the mice.
To see if there were parasites hanging around somewhere that didn't quite get treated.
Exactly. Because I want to know if they could get rid of all the parasites with these treatments.
That's another trick that they use for toxoplasma experiments as well.
So you would like to know the answer?
I would, of course. Everyone would like to know.
So no parasites detected.
Good.
Even with immunosuppression.
Excellent.
It looks pretty good,
yep.
right? And so they conclude that the
good.
3 milligrams per kilogram of this number 7 drug gave the best result with 90% cure in all animals surviving to sacrifice.
So how heavy is a mouse?
How heavy?
Yeah.
It's not. It's 100 grams.
Less. 30, 40.
Times 3 milligrams.
Well, per kilo.
Oh, it's even less, yeah.
Much.
It's not a lot of drug.
No, it's like microgram levels.
But how much would you have to take? Oh, never mind. We don't want to talk about your weight.
Less and less as time goes on because I've been losing weight, I hope.
So that's cool. Even after immunosuppression, no parasites.
Not bad.
Wipe them out.
Not bad.
So they said these studies establish a proof of concept.
Right. Good.
For nitrile-based inhibitors. What do you think of that?
Of cruzipain. I think it's great. Now what's next?
Nothing.
No.
Because Merck Frost is gone.
No, come on. They're gonna sell that drug to another company that can do the right studies. What would you do next, Vince?
What would I do next?
Yeah, what would you do next? If the money were not the object and you had a company of your own and you knew this compound existed, what would you do next?
So the FDA in the U.S. says you need 2 animal models to
Two.
So you need another one.
Right.
Is there a non-human primate model?
Of course there is.
Too expensive, though, right?
Yeah. In fact, the U.S. is about to disengage from research on chimpanzees.
It's done it, yeah.
There are 360 some odd chimpanzees out there in laboratories that are about to be released.
They're going to let them go?
They're going to take them to a special place.
Oh, yeah. Where they'll be cared for, yeah.
Yeah, until they die of old age. That's right.
And they can talk about their research experiences.
I guess. Yeah, right. That's right. Oh, I was a control. Yeah, I got the placebo. I didn't, I had an easy life.
But you can use macaques.
Could.
Just some other model to
Aotus.
show. And they also have to do a lot of other toxicity experiments to make sure it's not toxic.
Any other animal you wanted. You could use armadillos. You could use whatever you'd like because Trypanosoma cruzi infects all of them. You could use dogs.
So the thing in this Dickson is, which I didn't mention, these inhibitors, when they were doing the in vitro studies with the purified enzymes, they also used some cathepsins, and they're not very selective.
Right.
So the mouse, they didn't really look for toxicity. They just looked for survival.
Right.
And these mice could be suffering from a variety of issues. So, for example, they could have liver problems, but they don't know because they're not doing liver enzyme studies. So you need to do a lot of toxicity.
There's some other studies that you need to do too, don't you? Like biodegradability. What happens if this drug is so resistant to the P450s and to other categories of degradation pathways that whatever goes in comes back out again?
Well, we know that the kinetics are not consistent
But we
with
don't
that.
want this drug to enter the ecosystem and have its effects on other life forms, do we? No.
Yeah.
So that's another issue. You have to look for it in feces and in urine and in sweat and all kinds of other things too, right? So you got to really be thorough about this one.
I'm not sure that these compounds are the ones to bring forward or not. I suspect you might want to modify them to make them
So
more
you're the company.
active.
You're going to say, okay, I've got some parent compounds, and it's got a certain degree of toxicity, and we've done some studies on the R groups, right? And we know that this R group is more toxic than this R group, so we'll change it, and we'll try to modify it, and we'll try to make it more selective, and we'll try to make it more bioavailable, and we'll try to make it more soluble, because you have to give this in some way too. And then we'll try it in, and now you have to fill in that blank. What will you try it in for your final proof that this is a good drug to try clinical trials with?
So, in fact, here they say in the discussion, a new inhibitor, number 9, with an excellent selectivity profile has been recently identified and studies are in progress. You modify the compounds in ways to improve their properties, and then you repeat these mouse experiments. You do another animal, you do toxicity, extensive toxicity in cell cultures and in animals.
And
then you can do a phase 1 if everything looks good.
You go to people.
What is phase 1, Dickson?
Phase 1 is just tolerable or not tolerable.
Safety, mainly safety.
You give it to people that don't have the infection and see what happens.
Mm-hmm. Yeah, make sure they're okay. And you
Remember,
measure...
You're forgetting that Charles Knirsch is a very good friend of mine, and he was
you never talked
vice
to him.
president of Merck, or Pfizer's anti-therapeutics. So I've had a belly full of... and not enough. I mean, I could learn more, of course, but I talked to him a lot about how drugs are developed and tested and that sort of thing. So I'm pretty familiar with that. And Bill Campbell is a good friend of mine too, so he's told me all of that as well. So you've lived with one for most of your adult life, and so you're giving me the rhetoric from industry, and I appreciate that rhetoric because that's the way you should do it. So now you've got all of the studies that are finished, and you're ready to start your clinical trials. Who do you start with besides the safety issues? Safety issues, that's fine. Who's your first clinical trial group?
You know the answer.
I do. I have an answer here. I do have.
You're going to go to South America?
You have to. That's where the
I
disease
don't think
is.
you always have to.
No, but you'll have to
For
go
phase
there.
1?
Yeah, you have to go there.
But trials are not always done in the population that's getting the drug.
But you have to go there because...
What?
Because that's where all the sick
Yeah, I know
people
that,
are.
but for toxicity, those...
No, we've already done
But
those.
they're genetically different.
We did
I
that.
agree.
We did that already. I said
you're in
we
phase
did that
2
already.
now. Okay.
Phase 2.
I don't know. It really depends, Dixon. I would guess, the best population is where you're going to use the drug.
Well, you take the sickest people that have no hope of cure otherwise.
Are you going to take... Here's the thing. Are you going to take people who are already sick and treat them? Or are you going to treat a population and look at the incidence of infection?
No, you can't give this drug that way, I don't think.
This is not a preventer. This is if someone has already got the Roman eye. They come in, you're going to treat them with the drug, right?
No, I wouldn't treat those people with this drug yet. I would save those for clinical trials number 3.
What are you going to do in 2? You got to have a dosing regimen.
I'm going to take the sickest people that have no hope of cure otherwise, and I'm going to get permission to give them 3 different doses of this drug.
Mm-hmm.
And I'm going to watch to see what happens. And when one group starts to do better than the other 2, I'm going to switch them all to the group that does the best. And I'm going to start taking tissue samples, and I'm going to start taking measurements of their megacolon and megaesophagus and the megacardia symptoms. And I'm going to see whether I can reverse them or stop them from progressing using this compound. And if it works, okay, if you're lucky, and it's a well-tolerated drug, and remember, these people are sick already, so even if it has no toxicity effects whatsoever in normal people, it could still have a toxic effect in somebody who's compromised by the infection. So you might get toxic effects that you would never see otherwise.
Mm-hmm.
So that doesn't mean that just because you did the right studies, it isn't going to result in something bad. All right?
Of course.
So, okay, so let's just, under the best of circumstances, assume now that this compound clears that hurdle and you've actually affected a cessation or a reversal of effects on, let's say, the cardiac tissues, which can repair themselves, by the way. Okay? You know what the leading cause of heart transplants are? Chagas disease. It's interesting, isn't it?
Not in the U.S.
Of course not.
U.S., it's probably some viral infection.
Sure. Or some genetic defect.
Right.
But in the...
Or supersize me.
No, that's another kind of...
Diet, I mean.
Yeah. If you go to South America, the leading cause of heart transplant, the reason for doing a cardiac transplant is to cure somebody of Chagas.
Okay.
So if that works, then you would look around for a hot spot for the transmission of this infection. Now, where do you think those are?
South America.
No. In South America. Where do you think they are?
Brazil. Argentina.
Be more specific.
I don't know.
Well, is it in the cities? Is it in the suburbs?
No, it's in the suburbs
Is it
because in the cities,
jungles,
the bugs don't bite your face.
Where are the
because you have
reduvid bugs
screens on
found?
your window.
Where are they found?
Let me look it up. Reduvid rain. What do you call where a thing lives again? Reduvid habitat. Reduvid habitat.
And there are many different species, by the way.
The reduvid bud is found largely in Fort Lee. Did you know that?
Because I have a secret colony growing there. Did you know that post-Katrina, southern United States...
Reduvid bugs are... Sorry, yes.
They had an outbreak of trypanosomiasis.
Because the bugs were blown up from South America?
No, everything was damaged, and lots of things didn't survive.
I see. But they lived there originally, right?
Yeah, that's true.
Reduvid bugs are found everywhere, from the southern U.S. down to Argentina.
This is correct.
But here they're showing pictures of mud shacks.
That's right.
Don't they live in the roof of the mud shack and fall on your face at night?
That's part of it. They don't fall on your face. Their defecation products fall on your face.
Reduvid bugs hide during the day and come out at night to feed.
So
they are attracted to warmth and carbon dioxide.
Well-developed... Let's try this again.
You know they could bite for half an hour?
Hang on. Yeah, they can, because you're not going to say anything because you're asleep. So this is mostly a rural infection.
It is.
That means you have to use this drug in a rural setting. That's tough.
Look at this picture I found, Dickson. Yeah. You're going to freak out.
Oh, no. I listen.
It's a bag of them.
Bob Gwads used to bring them all up with him when he taught the course with me on the vectors.
Look like stink bugs.
He had live reduvids of various stages because they undergo an incomplete metamorphosis.
You know what some of the aliases are for reduvid bugs?
Kissing bug, cone nose bug.
What else? Chinches.
Rodnius prolixus is
assassin
one of them.
bug.
Assassin bug.
It's a nice name.
It's a nice one.
Assassin bug.
Yep.
Do you want to do some email?
Wait a minute. We're not done.
We're not?
I'm going to raise this question for you.
Oh, okay. Go ahead.
Let's say it's a perfect drug.
Yeah.
Where are you going to use it?
South America.
No, that's the wrong answer.
Sorry, I'm sorry about that.
The people who live in cities, you don't have to worry about them because they're not catching it there.
Of course, rural South America.
Who's going to dare go out there and give this drug? And how would you know who to give it to?
Well, are there physicians in these areas?
Sometimes yes, sometimes no. Remember I showed you a picture of mucocutaneous leishmaniasis?
Yeah, it's horrible.
How could anyone go that long without medical attention?
Well, it's part of the
So
issue.
that answered the question.
That's a whole nother
They
issue.
ain't no physicians out there.
All right.
There's none.
So you're saying don't even bother with the
No,
drug?
I didn't say don't bother.
You're
but
saying
I'm just
develop
saying,
health care in South America, rural areas.
There's another approach to this problem.
What's that?
And that's to control the reduvid bugs by not building thatched houses.
Well, that's not happening.
So Joel Cohn, do you know who Joel Cohn is?
No.
He's over at Rockefeller.
Is he still there?
Yeah, sure. He's at the Population Council in Rockefeller. And he once got involved in a project in South America to redesign rural housing to prevent the establishment of colonies of reduvids.
Okay.
And it worked, okay? Wherever they did it, wherever they made these cement and cinder block houses with no thatched roofs, the reduvid bugs could not be found near the houses.
Is that a low-tech solution?
It's not so low-tech.
Because,
actually. It's pretty expensive.
filtering the water helps dracunculiasis, right?
Yeah, it does, but that doesn't go on too much either.
Low-tech screens in the windows.
Yep. No, that's
Are
right.
you saying that you would rather do
Mosquito
that than
nets.
use this drug?
Nope, I didn't say that at all. But a lot of, well,
Who's going to pay for this drug?
That's right. That's another thing. Yes, the answer is yes. Remember, who's on first? No. Who's in Geneva?
WHO.
That's
But
right.
they don't pay for it. They get donors.
Well, they do have an orphan drug program. And this might fit into the orphan drug program. Okay, let's read some
Do you want to do some email?
Just raises the questions.
No,
that's
I'm
all.
sorry to presume that you were done. But you always can interrupt me. You know that.
And I'm not interrupting. I'm just adding to the mix, as it were.
First one is-
since I don't have a screen in front of me, you're going to have to read them all.
Well, you know, you're always welcome to have a screen.
No, I know,
Do I
I
need to
know.
buy you a permanent screen for
No,
this
I have
room?
my laptop out there, but I just didn't bring it in this time. I'm neglect. This is our first edition after the new year, and I'm just getting back in the swing.
First one is from Andre, who writes, "To my great delight, I just discovered your podcasts, TWiP, TWIM, and TWiP. The first TWiP I heard about Strongyloides stercoralis, although informative and interesting, seemed to have several inaccuracies. I was not able to find Dickson's email, and therefore I am writing to you."
Uh-oh. So I have to get on the defensive now. Go on. Are you going to put that down on top of my book? What if you...
Dick said that Strongyloides, when it enters the host, becomes a protrandrous, he's written,
well, it's not
hermaphrodite
Yeah, go on.
adult. Actually, this is not true. The parasitic stage of Strongyloides is a female that reproduces by parthenogenesis.
And what is his reference for that?
I'm going to read you the rest. It's a long email. Hang on. He also said that a protrandrous nematode is first male and then becomes female so that it can self-fertilize. Actually, as I said,
Right.
Strongyloides is not a hermaphrodite and therefore it cannot be protrandrous. The only nematode parasites that I know to be a protrandrous hermaphrodite are the worms of the genus Rhabdias, which usually infect amphibians and reptiles. C. elegans is a good example for a protrandrous hermaphrodite nematode. The term protrandrous means that the organism first produces male gametes and later shifts to the production of female gametes. The only difference between C. elegans, hermaphrodites, and females of closely related species is the ability of the hermaphrodites to produce a limited amount of sperm just before becoming adult. These female-bodied C. elegans with sperm are not considered males because they cannot transfer their sperm to other individuals. The sperm of these worms are stored in the spermatheca and used for self-fertilizing the oocytes produced during adulthood. C. elegans males are morphologically very different from hermaphrodites, produce only sperm, and have specialized mating structures used to find and fertilize the hermaphrodites.
I sit corrected.
My field of specialization is actually evolution of modes of reproduction of nematodes.
Oh, wow.
I study a free-living nematode that can produce males, females, and hermaphrodites. In many ways, it resembles parasitic nematodes because larvae that sense the lack of food become dowers, which is the dispersive stage of free-living nematodes. Dowers are the equivalent larval stage as the infective stage in parasites.
Here, here.
Dowers can resist harsh environmental conditions, crawl in arthropods, and be transported to new places. For the species of nematode that I study, the dowers develop into self-reproducing adults once they encounter a habitat with food. This makes sense because they can then colonize new environments without the need of a mating partner. The larvae that grow in an environment with enough food develop into females and males. And if you're interested, he gives a link to his work.
Where is he from?
He is from... the University of Texas at Arlington.
Cool. I had a good friend there once.
He's in the Department of Biology.
George Stewart and I were good friends, and he was there for a long time before he moved.
And he works on this worm here, Rhabditis species. That's what he works on right there. See that?
Yes, I have.
So we will put a link to that. Evo Devo of squamates. You know what Evo Devo is? No?
Sounds like a singing group.
What is Evo Devo? This is a new field. And you know who founded it? Cliff Tabin, who I was with at the Cancer Center.
All right.
Evolutionary Development.
Got it.
Evo Devo.
Okay, so that's the beauty of this show.
Evolutionary Developmental
Sometimes
Biology.
You can say things that are wrong, and other people will correct you, and now our listeners are fully informed. And I have no...
You look sad, Dickson.
No, I'm not sad at all. I'm happy to be corrected.
I'm corrected all the time. Don't worry.
No, I'm not.
Okay.
I'm not worried at all.
So the downside, I'm sorry, the upside or the...
Yes. The
take-home message is that Strongyloides is not a protrandus hermaphrodite.
Okay.
Noah writes, I have been a huge TWiP, and TWiM listener for about a year. I love the show, TWiP, the most. I love listening to you and that Deponia guy.
Deponia.
Deponia.
That's me.
You guys make me laugh, but more importantly, I have attained a great deal of knowledge from the podcast. Can't help but tell my colleagues about the amazing things you two talk about, from toxoplasmosis or parasite epidemics.
Good.
I'm writing to you because tomorrow night I'm going to post you as the American of the Day in my Facebook group because of your great accomplishments and devotion to educate the public about science. So this is back in October.
Highly honored.
And I will put a link to that. Thank you for that, Noah.
Great.
Devin writes, so Devin is my son's name, but this is not my son.
Not your son, right?
Hey, guys, I'm from the San Francisco Bay Area, and I wanted to extend my appreciation for the show. I listen to both TWiP and TWiV and would be disappointed if you didn't produce the show into the future. The conversations require the listener to pay attention a bit more than the average podcast, I believe. This may in part be due to the use of terminology that I often find myself looking up during the podcast or later when I've heard it more than once on the show.
Got it.
As a result, I feel like I'm in on a conversation that I normally wouldn't be, and this inspires me to try even harder to understand the concepts.
That's wonderful.
One more thing. While out at a bar the other day, I had randomly met someone who had his PhD in the biosciences.
Right.
I don't remember specifics. I studied Earth and planetary science. And I had mentioned that I listened to TWiV and TWiP, and he knew who Vincent was, which led him to add your podcast to his iTunes at the bar.
Cool.
Keep up the good work. I looked for your podcast page on Facebook and didn't find it. Did I miss something? If not, you should create one. It's a good way to promote your show.
Do we have one?
Remember, for every person that writes an email, there's probably a bunch more that want to, but just don't get to it.
That's
Thanks,
true.
guys.
I agree with that. That's totally
Facebook.com/thisweekinvirology. I put everything there. TWiV and TWiP and TWiM. Facebook.com, This Week in Virology, all one word. We have over 6,000 likes.
Nice.
It's pretty cool. You go to a bar and you meet people that know Vincent.
Hey.
I think that's a great story, Devin. All right, John writes, Dear Professors, it's 1 degree outside my window as I write 3 comments about TWiP 63.
TWiP. Okay.
1 degree C or F?
Exactly.
Probably C, right? Exactly. Because he knows we write it in C.
Where's he writing from?
He doesn't say. 1. You wondered how plasmodium DNA ended up in ape feces.
Yeah, I did, actually.
I learned biology when life was made of cells rather than molecules. So I think of the body not as a big amino acid sequence, but rather as a series of tubes. With this old-fashioned background, I thought the path dismissed by Dickson was obvious. The liver recycles old red blood cells to recover iron. It dumps the rest of the cell contents into the intestines. If it recycles an infected red blood cell, the waste may include plasmodium DNA. That's what I suggested, and you poo-pooed it.
I poo-pooed? No, the chimpanzee poo-pooed it.
That's good, Dickson.
Thank you.
Yeah, you get off today.
Do I? Excellent.
So
I was just curious as to why it would survive all the way down through the gut tract, considering all the DNA activity that you might encounter and stuff like that.
Should I ask...
yeah. Beatrice
Hahn? Why do you have a better memory than I do?
I don't know, Vince. I'm much older than you are.
You're in pretty good shape.
I feel okay.
Except, yeah, you are in good shape. You're not dizzy at all.
Not even a little. But although by listening to me, you might not say that. You might say, that guy's dizzy.
No, I never say that. Yeah. You're a little like Luddite, but you're not dizzy.
Not Luddite. I'm just
You don't like
cautious.
technology.
I'm cautious.
You don't tweet, you don't Facebook, you don't Google Plus.
I don't.
You don't see the value of social media.
I don't.
You don't have anything to say. All right, give me that. Number 2, how do merozoites know when to stop producing copies of themselves and make gametocytes instead? I can imagine 3 mechanisms. A, random chance. B, a molecular clock like telomeres, which allows cells to count generations. Or C, environmental cues related to overpopulation, such as oxygen concentration. If you give me a grant in a lab, I would be happy to watch a team of grad students investigate.
I have a feeling it's already known, and I just didn't know what it was.
That's
but
funny.
We can try to look it up. That's a great series of suggestions. So
you would agree with them?
Oh, well, I would at least consider those as possibilities, sure. You bet.
Okay, and finally, 3, do I understand correctly that a deadly infection can result from just one Plasmodium sporozoite cell lodging in the liver 15 minutes in? That's when you said that.
Yeah.
Is that true? 1?
I believe that the number of sporozoites needed to initiate an infection varies from 1 to 10.
Should we ask an expert?
Robert Quads would know the answer.
Where is he?
He's at NIH.
Can you email him and ask
So
him?
we could call him right now if you want.
I don't telephone.
Hey, yo, you know what? You're talking about me as a Luddite. You don't telephone. What are you talking about? That was one of the oldest communication devices besides a drum.
People don't use the phone on their smartphones anymore.
You can say that all you want. I use it all the time.
You're a Luddite.
No, I'm not. I have a smartphone.
I want to get through the rest
I even
of
put my
these.
iTunes on my
Scott
smartphone.
writes, gentlemen,
Stop bickering.
as a resident of Latin America, it was with some interest that I noted this story in Science Daily.
Yes.
I would appreciate your comments.
Okay.
And he sent the Science Daily article on the paper we did today.
Ah, cool.
New drug candidates show promise for cure of Chagas disease.
Very cool.
I would like to know if the parasite is a threat to the 2 cats. I'm sorry, no. As I have on several occasions discovered the insect vector in my living space, this parasite is of some concern to me.
It should be.
I wonder where he lives. Oh, he lives in Costa Rica.
Costa Rica.
I wonder where, though. Cartago. I don't know if it's rural or epi-rural.
Was it near San Jose?
Mezzi para-rural.
Mezzi para? No, stop it. What was that trypomastic that you said before?
I'll have to listen. I don't remember. I would like to know if the parasite is a threat to the 2 cats who live in the house with me or my dog who lives in my yard.
If they eat the reduvid bugs, which they are prone to do when they find them on themselves, the answer is yes. That's how dogs catch it.
You said it infects everything, right?
Yeah, it does. It does. They are.
I have not been able to find any information on the veterinary implications of this parasite.
They are. Oh, there's lots of data on that. Lots of it. Go to PubMed. Type out epidemiology of Chagas disease in domestic or peri-domestic animals, and you'll find a lot of data.
Well, I just searched for Chagas in pets.
Yeah.
And there's a website, petmd.com, Chagas disease in dogs.
Tons of it.
There are PDFs from Iowa State.
Oh, there's a bunch.
Texas A&M.
There's a bunch.
Just search for Chagas in pets on Google.
Or Chagas in peri-domestic animals. You'll find it.
Scott in Costa Rica.
Yep.
Neva writes, hi, Dickson and Vincent.
Mm-hmm.
Neva's our friend from Buda, Texas,
Like it.
I guess the TriCaster medical device is almost here. If this is viable, very cool. What's next? And she sends a link to an article. Rice University researchers have developed a non-invasive technology that accurately detects low levels of malaria infection through the skin in seconds with a laser scanner.
It measures hemozoin.
The vapor nanobubble technology requires no dyes or diagnostic chemicals, and there is no need to draw blood.
Yep.
You've heard about this?
Sure I have.
I got a 404 error. I'll have to look for the right
No, I've
link.
heard about it. Hemozoin is a byproduct of their metabolism. Remember when they had the stacking enzyme inside the red cells, and they eat the globin part?
Yeah,
and
I
they
do
throw
remember.
away the heme?
Yes.
And they're the enzyme that actually allows the heme to be deposited in the middle of the parasitophorous vacuole, where they all are developing in synchrony, forms this layered compound.
Yes.
When they pinch off and all go out of the cell, they leave this behind, and then the cell disintegrates afterwards, releasing the hemozoin into the blood. It has a specific spectrophotometric signature, which they can actually detect by looking at the blood that circulates at the skin level.
So basically, all right, so this is one test. We'll be able to do this for many, many diseases at some point, non-invasively.
Probably.
Why is it good to be able to do this, Dickson? Non-invasive detection of malaria.
Because you can go in the field and just have little kids walk past this machine. They wouldn't have to get stuck. It wouldn't hurt them.
And if they're positive, what do you do?
Then you start treatment.
Who's going to pay for this?
Yes, they're in Geneva.
Okay. Yes.
No, they're on first.
Never miss your great podcasts. Neva from Buda.
Glad to hear from you again, Neva.
Remember Neva?
I do.
Are you just saying that?
Neva was a nurse?
No, I don't think so.
No, we have another
that's
nurse that
from
listens
Naples,
to us.
Florida.
Oh, that's okay. Neva is not in Naples and not a nurse.
Neva's not
so
in Naples and not a
Neva's
nurse.
never...
Very good.
I wonder if she's into nanotechnology.
Apparently. Vapor nanobubble.
Neva,
That's the name of this technology.
Neva Nano. Sounds like a Mork and Mindy type of thing, right?
Now you're aging yourself.
Nano.
Never miss your great podcasts. I already said that.
You did.
The last one is from Jay,
Okay.
who writes, "Hello. I got recommended your podcast from a colleague. I just wanted to know what song by RJ do you play in your intro.
You know that.
I emailed him and he thought it was guitar sounds, but it's not quite right. Do you mind posting the title somewhere? Thanks.
You can do it.
The intro is called Halloween Remix.
Aha.
The outro is called Guitar Sounds.
Nice.
So the outro is Guitar Sounds, but it's not the intro, as you
Very
suggest. It's a Halloween Remix. TWiP can be found at iTunes and also at microbeworld.org/TWiP. If you like what we do, well, we thank you.
We do.
And we'd like you to go to iTunes and star the podcast and leave a comment leave a comment.
You bet.
Because that helps to keep us visible.
It do. I mean, it does.
Are you tired? You had enough?
I'm not sure I'm tired or not.
Almost 2 hours.
I've been back from India now since Sunday, and we didn't talk much about that, but so I spent 2 weeks in India with my wife at a meeting in Chennai, and got back Sunday morning at around 6 in the morning, and at 8 o'clock in the morning, they closed Kennedy Airport because of the fog and the ice on the runway. We missed being diverted to, like, Miami. Too bad. By 2 hours, just by 2 hours, we missed sliding off the runway. But I had another thought here that okay, we'll get to it when you start telling the listeners about our websites and where we can be found.
So after this, we're going to look out the window and see what the ice flows are doing.
We are.
Because we videotaped
They're
them
going
this
that
morning.
way now.
We're going to see if they're going up the river.
They are going up, I can guarantee you.
And we're going to make a movie. I'm going to put it on YouTube.
They're going up because the tide has changed.
What did you call them? Ice cakes?
No, pancake ice.
Pancake ice. That's going to be the name of the video.
Pancake ice.
I bet there's already one there. But
maple syrup.
Pancake ice and TWiP.
That's right.
And you can always send us your questions and comments.
Yes.
TWiP@twiv.tv.
We can.
Please send them to TWiP@twiv.tv. That makes it easier for me to sort them out because we get a lot.
You bet.
I'm not complaining.
No, of course not.
But I'd like help in sorting.
There you go.
Dixon Despommier can be found at
Now, if you're going to say Trichinella.org, we're going to have to change that now.
Yeah, because it's dead.
It's not dead. It's offline, and I'm donating the website to the International Commission on Trichinellosis.
And they're not going to do anything with it.
You know they're going to remassage it into a larger, more generic website that will cover all the issues that were covered on my personal website for Trichinella, because I no longer do work on the organism. And they will add some things to that, because they were very positive and complimentary about the quality of the website. They love it. But they also wanted to change some things because it had fallen into disrepair. We didn't water it enough to satisfy their
they're right.
needs.
You hadn't updated it in a long time.
No, that's true, because I haven't worked on Trichinella since 1999.
Right. Well, medicalecology.org is also
Still up.
It's up, but it's not been updated in years.
No, but it's still got a lot of great information.
It's got info.
It's got lots of
info. And then, of course, there is...
Well, yeah, there's a verticalfarm.com.
Which gets updated once a month. Right?
Well, probably not as much as it should. But we're working on an interactive map right now, so that should be good to identify where all the vertical farms in the world
That would be cool.
With their websites.
Who's going to pay for that?
That's a very good question.
They're in Geneva?
Yes, the answer is yes.
Wow.
Yeah, we want to do that next.
Thank you, Dickson.
You're welcome, Vincent.
You look really tired. You should go to sleep.
Thank you.
Just kidding.
For that. Dizzy.
You're also at the Huffington Post.
Yeah, but I've only done one of those so
what's
far.
the story? Why don't
I'm
you write?
waiting for something to write.
You said the other day you had something to write.
Yeah, I have to write something about hunger. I just saw some stuff in India.
All right, let me give you a suggestion.
Please.
You're out there, I see you. You're 10 feet away from me. You talk most of the day to other people. You need to focus, chow down, and work.
I'm working. You don't
Too many distractions.
That's true. I should stay at home and do this.
No, you don't have to stay home. It's actually better to get here and get out of the house, get a new environment.
Yeah.
But you need to focus and stop talking to people so much.
You think?
No, I'm just kidding. Talking to people is what makes you Dickson Despommier.
Well, that's what makes my life interesting at this stage of my career, that's for sure.
I'm Vincent Racaniello, and you can find me at virology.ws. The music at the beginning and end of TWiP, as you heard, is composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. He has a brand new CD out. I don't think you'd like his work, Dickson.
Why do you say that, Vince?
It's modern.
Do you know who Pat Metheny is?
Yeah, Pat Metheny Group.
You do, eh?
Yeah.
Think that's modern?
Oh, he was around when I was a kid.
Do you think that's modern? He was not.
Yeah, Pat Metheny is old.
He's not.
Absolutely.
No, look him up. He's only about 40.
When I was a kid, Pat Metheny...
No, you're younger than I am. You're younger
was
than I am.
Playing.
You're younger than I am.
So I wouldn't consider him. Founded in 1977.
Okay,
it's a jazz group. I do like Pat Metheny.
He's great.
But you don't like Pink Floyd.
I do so.
You said yesterday you did.
No, I didn't say I didn't.
Maybe that was Steve Goff.
I love Pink Floyd. I love it.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.